Hello, everyone, and welcome to today's webinar, The Value of IgG Testing for Food Tolerance and Intolerance. My name is Amy Wilson. I'm a Senior Product Manager here at UPCO, and I'll be the moderator for today. UPCO is pleased to be the exclusive distributor of the MacroArray Diagnostics multiplex IgE and IgG testing platforms in North America. I'm also pleased to have with us today Dr. Sandra Wieser. Dr. Wieser is the Product Manager for the ALEX IgE and FOX IgG testing platforms at MacroArray Diagnostics. Before we get started, I have a few things to note. First, today's presentation is being recorded. You will receive an email within 2 to 3 days with a link to access the recording that can be shared with colleagues who may have been unable to attend today. Next, we'd love to hear from you during today's presentation. Please submit any questions you have through the chat box. If we can't get to your question during the presentation, we'll be sure to follow up, and during the Q&A, we'll be sure to follow up after the presentation. Finally, there will be a brief survey at the end of the webinar. We would appreciate a minute of your time for some feedback. Without further ado, I'd like to welcome our speaker, Dr. Sandra Wieser.
Thank you very much, Amy, for this very kind introduction and invitation to this webinar on food intolerance testing. My name is Sandra Wieser. I'm a product manager at MEDEX for nearly 2 years, and I'm really looking forward to guide you through the new era of food intolerance testing. I switch off the camera now, and we'll start the presentation. So a lot of people today are suffering from chronic diseases like abdominal pain, diarrhea, migraine, depression, skin problems, but also many other inflammatory chronic diseases. And they ask themselves the questions, why do I have all these symptoms? So several diagnostic procedures, examinations, and tests have already been carried out, but without diagnosis and suitable therapy. So they are already really frustrated and want clarity. For some of these patients, the answer could be in the gut. If you keep in mind that only 2% of the general population suffers from IgE-mediated food allergy, it's estimated that food intolerant affects even more, around 5% of the general population. I estimate that the number is quite higher because a lot of people do not connect their symptoms to food intolerances because the symptoms appear quite delayed. And also the general practitioners do not do generally IgG testing. They go for other examination tests. So I guess food intolerance the number should be even higher. If you keep in mind that the immune system of the gut is the largest in the entire body, you can already imagine how important the gut is. The intact gut barrier is hardly permeable. It's the key for tolerating food and being healthy. And under optimal conditions, it's almost insurmountable for bacteria, viruses, disease pathogens, and components from food. A lot of factors decide whether the gut is healthy or not. There are a lot of factors that can damage the gut epithelium. On the one hand side, there are unhealthy lifestyle habits like stress, alcohol, but also other factors can influence the gut, like medication, antibiotics, diseases of the digestive tract, inflammation, the leaky gut syndrome, but also infections with parasites, bacteria, and viruses can influence the gut epithelium. So to understand the difference between food allergy and food intolerance, I want to show you this slide because I think it summarizes the most important key features of IgE-mediated food allergy and IgG-mediated food intolerance. For food allergy, the symptoms appear quite immediately, within 2 hours after the exposure. But for food intolerance, the symptoms appear delayed after 2 to 72 hours. So therefore, it's really difficult for some patients to understand if they have migraine or skin problems, that it was connected to food they have eaten 3 days ago. For food allergy, the symptoms can be really worse. They can be life-threatening, including anaphylactic reactions. The good thing is for food intolerance, the symptoms are not life-threatening. The course of action for food allergy is to avoid the triggering foods, symptomatic treatment, take antihistamines, go for immunotherapy if it's available, and keep an emergency kit if already severe allergic reaction like anaphylactic reactions have been observed in the past. And for food intolerance, nowadays the only course of action is go for IgG testing and avoid the triggering foods. On the right-hand side, you can see a chart showing the symptoms. Like for food allergy, for example, the gastrointestinal tract and also the skin is involved. But for food intolerance, the whole body, really unusual, I would say unusual organ systems like the brain, the mind, the joints, the muscle, the skin can be involved. So depression, headache, migraine, fever, sweating, weakness, and so on. These can be all symptoms that can be connected or can be the consequence of food intolerances. What causes food intolerances? For IgG-mediated food intolerances, it's not that clear than for, the pathomechanism is not that clear than for IgE-mediated allergy. But there is a theory called the leaky gut syndrome. On the left side, you can see a healthy gut. The gut cells are intact, the tight junctions are perfect. So the antibiotics, pathogens have to stay outside. The food components are taken up, digested, and enter the bloodstream, and tolerance is induced. On the right-hand side, you can see a leaky gut that has been destroyed due to different factors that I already mentioned before. And the gut cells itself can be damaged, but also the tight junctions in between the cells. So a lot of pathogens can enter, but also food components are taken up or enter the bloodstream that are not properly digested or even undigested. And there they get recognized by the immune system, and IgG antibodies are produced. And those IgG antibodies adhere to the food proteins, forming a huge complex, and other cells of the immune system try to eradicate, to destroy this huge immune complex, which leads to a lot of inflammatory reactions. And there's a theory how food intolerances and other diseases, other inflammatory diseases of the gut can occur. So the food intolerances can influence the whole organism. So the idea of Maddox was to establish an ELISA-based in vitro IgG test to detect those IgE-mediated food intolerances, and then enable individualized diet recommendation, and upon elimination diets, then the symptoms can improve, and this can lead to the well-being of the patient. This is just a short introduction because I now would like to guide you through 2 studies, and afterwards I will give you a lot of details of the FOX test. So this is the first study I would like to share with you. It's performed with 150 patients suffering from irritable bowel syndrome, and IgG testing was performed, and then the study group was divided into a true diet group and into a sham diet group. The true diet group had to exclude all those foods to which they had raised high IgG antibody levels, and the sham diet group had to exclude the same number of foods, but not to those where they had raised high IgG antibody levels. And the authors concluded from the results that the true diet group had a 10% greater reduction in the symptom score than the sham diet group. Similar results were seen from a study group where they analyzed migraine patient. It was a double-blind randomized crossover trial where they analyzed migraine patients. They did IgG antibody testing, and additionally to the elimination phase, they had to do a provocation diet where they really had to consume those foods with high IgG antibody levels. And for both study groups, they could see that there was a statistically significant reduction in the attack count, in the number of headache days, in the days where they had to take acute medication, but only during the elimination diet period. So there are a lot of migraine patients that are therapy resistant. And the authors from this study concluded that diet restriction that is based on IgG antibody testing could be an effective strategy to reduce the frequency of migraine attacks. Patients are frustrated, as I already said before, and they really expend large sums of money on a variety of unsubstantiated tests. And therefore, IgG-based testing has been proven useful as a guide for elimination diets with clinical impact. So go for IgG testing, and then exclusion diet, elimination diet, rotation, whatever, and then the symptoms should improve, and this should enhance the quality of the life of the patient. A lot of elimination studies exist that have been shown that upon IgG testing, this can really improve the patient's symptoms. So now let's come to the FOX, to the Food Xplorer. Today, it's the most comprehensive IgG intolerance test. It's a semi-quantitative in vitro diagnostic tool to test 286 food antigens from 13 food groups. It determines the concentration of food-specific IgG antibodies in serum or plasma. It detects all IgG antibody subclasses 1, 2, 3, and 4, and only 10 microliter of serum or plasma is needed. It includes, additionally to the basic foods, also superfoods and novel foods. This is here a chart showing the 13 food groups that are included. So we have vegetables, fruits, seafood and fish, cereals and seed, egg and milk, meat. We have edible mushrooms, novel foods, a lot of other foods, coffee and tea, nuts, spices, and legumes. So these are the three key features of the FOX. It offers the most comprehensive antigen panel. It contains superfoods and it contains unique foods. This is here a comparison of a FOX with another competitor on the market, and we wanted to see which foods do the other companies offer and have, and we could see that we are the only one that offers novel foods. The other brand does not have any novel foods on their IgG test. This is here a picture showing you the superfoods that are included. So we have coconut, we have physalis, vanilla, cocoa bean, maca, mulberry, guarana, and we have more than 50 unique foods that are only available on the FOX test. For example, we have a lot of milk alternatives like coconut milk, almond milk, but also a lot of new fancy foods like moringa, baobab, that can be taken up via food supplements too. Nori, yeah, the list is quite long. I just wanted to summarize the most important ones. So more than 50 foods are just unique on the FOX test. To understand the technology, how it is performed, so in this example, I show you the banana extract, and the banana extract is in the first step coupled then to the nanoparticles, and in the second step, these nanoparticles with the antigen extract on the surface are then coupled to a solid phase, as you can see here. And if you want to do a FOX assay procedure, the first step would be serum collection. So you take blood and then you prepare the serum. The first step is serum incubation, a washing step. Then the second step is detection antibodies added, further washing step, and then upon the enzymatic color reaction, an image is taken, the results are analyzed, and you can look at the report. This is how the lab report looks like. On the left-hand side, you can see a summary where you have an overview of the IgG profile. You can already on one view see that there is a color code. So green is a low IgG level from 0 to 9.9. Yellow is from 10 to 19.9. And then in red, it's a highly elevated IgG level, also above 20 microgram per milliliter. And then if you want to go further, you can see all the different milk and eggs that were analyzed, also the cow's milk, the molecular allergens, and so you can go through all the details. What we also offer is a multiplex on demand. So if, for example, a doctor or a dietologist just wants to look at the nuts, then you can just hide all the other results. They are not deleted, they are still there, but you can just look at the nuts. And there are predefined MOD panels, but you can also, of course, do your own. And finally, you have a FOX lab report where you see all the tested food sources, and we have also a Raven for FOX, so you have an interpretation including alternative foods that can be consumed instead of the food with high IgG antibody titers. This year was done in our company with serum from myself. We just wanted to see how how the FOX behaves if we compare it with another test, and we could see here that the FOX test, there was a match in 12 foods. We were just able, of course, to compare those foods that were present on both multiplex intolerance tests, but for us it was quite interesting that 25 foods were only positive in the FOX test but negative in the other test, and only one food was negative in the FOX test and positive in the other test. Of course, when we did the FOX test, we had to do a lot of performance characteristic, and this here is just to show you that we compared the FOX with another test. It was the rider chip from our biofarm, and there was a mean qualitative agreement above 80% for the majority of priority food extracts. There was an excellent correlation with other IgG testing methods. To study the precision, a lot-to-lots variation studies have to be performed, and all of you know the lower the CV, the better it is, the better the precision is, and we could see it's comparable to the Alex test or a lot of other antibody tests. Is if you have higher titers, if you have a high IgG titer here, the CV goes down. So you have a quite good correlation or good precision already if you have results 10 to 19, but it gets even lower if the IgG antibody titer goes above 20. Then you have around 6, 3, 4. It's a really good precision. So it's a really accurate test. To study the repeatability, we did repeatability studies with multi-sensitized samples, and also here, the higher the IgG level goes up, the lower the CV. So it's really quite low total CV, and the results were really reproducible. We were also interested if there are interfering substances like heparin, citrate, EDTA, for example, but they do not interfere at all. So you can use normal serum, heparin, citrate, EDTA plasma. We were also interested if other components from the blood can interfere, like hemoglobin, bilirubin, triglyceride, and they had no relevant impact on the FOX test result that could be detectable. To study the specificity, we did cross-reactivity studies where different immune globulin subclasses were kind of coated in replicates on the membrane, and then the normal FOX procedure was performed, and we could see that no IgE, no IgM, and no IgA reactivity could be identified. So therefore, we could conclude that the FOX specifically detects IgG reactivities. The good thing is that you can use the FOX with the normal equipment, so it's fully compatible with an existing equipment and lab procedures if you have already the Alex test. If not, you can combine both tests on the same equipment. Also, the Image Explorer, the Max 9K and the Max 45K are compatible for FOX and Alex tests. Just a few information on the Image Explorer. It's our imager device for manual analysis. It captures an image of the array. You can scan one test cartridge at a time. It's integrated with Raptor server. You just need a PC with USB port and Raptor agent software. You need 10 microliter sample volume for the FOX test. The total assay time is 4.5 hours, but the hands-on time is 1 hour. To show you the Max 9K, it's our compact version of the Max 45K. It's also fully integrated with Raptor server. You can do 10 samples or 10 cartridges per run. It's fully automated. One run takes around 3.5 hours, so around 30 samples you can do per day. The total assay time is 3.5 hours, and the hands-on time is only 10 minutes. And all MEDEX test systems or all MEDEX tests can be processed, of course, in separate runs. So you decide, I do FOX or I do Alex, not in one run, but of course you can do both tests. And it is classified as an IVD others. For the Max 45K, it's a benchtop bench analyzer, also fully automated, fully integrated with Raptor server. You can do 50 samples or 50 cartridges per run. One run takes 4 hours, and the hands-on time is 20 minutes, and it's also classified as IVD others. Additionally, I just want to give you the information of our softwares that we offer. We have the Raptor server analysis software and the Raven interpretation software. So the Raptor server analysis software analyzes the results, and it can operate multiple Mac systems like multi-Macs. It's time saving. You have a lot of languages. You have your lab report. There is a data loss protection and so on. And it's also important, not only for the Alex, but also for the Fox, to have this Raven interpretation guidance software where you have really alternative foods. You don't need to look what can I offer instead of this or that. You have really a lot of information there. It's really time saving. Yeah. So this is always my last picture. What I like to show you is which picture do you want to see. And we always want to see the right picture, the clear, the comprehensive picture. And we think that with the FOX test really offers a comprehensive picture of IgG reactive food antigens. Yeah, it's your choice. So finally, I just want to add the publications where I raised a lot of information and the studies, what I showed you today. And thank you very much for your attention.
Thank you, Sandra. This was very informative and very interesting. Before we move on to our Q&A section, I just want to take a few minutes to summarize a few key points and present a few additional examples for IgG testing. So the FOX Food Xplorer detects specific IgG antibodies to 286 actually food antigens. Testing is completed with only 10 to 12 microliters of serum or plasma for the FOX test. That could be from venous blood or capillary blood. It's 10 microliters during the manual protocol, 12 microliters for automated analysis. Our current product detects all 4 IgG subclasses. So this includes the IgG1 and IgG subclasses that are associated with immune response and intolerance, and also the IgG4 isotype, which is associated with immune blocking function and tolerance. I'd like to share one recent example of the utility of IgG testing with one subject. In this example, the test subject was negative for IgE antibodies to all of the nuts tested in the FOX panel. But the subject was positive for IgG antibodies to both cashews and to almonds. In speaking with our subject, ingestion of cashews preceded GI symptoms. So that was a case of intolerance, whereas ingestion of almonds on a pretty routine basis caused no symptoms. So it's a case of tolerance. And it's just an example of how the total IgG results can be combined with actual food testing, experimentation to really determine if it's a tolerance issue or intolerance. In cases where IgG antibodies are positive for many foods, the addition of a specific IgG4 test may help to fine-tune the list for elimination testing. So in this case, if the subject has a high total IgG concentration with a low IgG4 concentration, we would expect intolerance. If the total IgG level is high, but the IgG4 level is also very high, we would expect that to indicate food tolerance. And a FOX IgG4 test to complement the current total IgG test is currently in development at Maddox. One published study highlighting the utility of IgG testing showed that increased levels of IgG1 and 3 were found in celiac disease, but elevated levels of IgG2 and 4 were more elevated in non-celiac gluten sensitivity. IgG testing can also play a role in the monitoring of immunotherapy. In a study of inhaled allergens, subjects with higher specific IgG to IgE ratios had fewer symptoms. In a peanut study, higher levels of IgG4 correlated with tolerance. And in another study, the natural resolution of milk allergy in children was associated with increasing levels of specific IgG antibodies. And whenever we talk about IgG testing, there are several points we'd like to stress. First and foremost, IgG testing is not a replacement for IgG testing. A negative IgG result is absolutely no indication as to whether or not someone could have even an acute response that's IgE-mediated. So IgE testing is still essential. Second, serological IgG testing is most useful when investigating recent issues, as antibody levels gradually decrease over time. As with COVID, where you have to get the booster after 4 months, if you have a response to a food, but then go for many, many months without having any interaction with that food, your antibody levels will decrease over time. Third, a positive total IgG result can indicate either tolerance or intolerance, so that needs to be linked to actual food responses. And lastly, some companies out there do offer specific IgG4 testing. One thing to note is that IgG4 testing alone may underestimate the potential for food intolerance, because you're not detecting those IgG1 and IgG3 subclasses associated with immune response. So thank you everyone for your time and attention. Thank you, Sandra. We do have a few questions that have come in. So one question, Sandra, we have is how the actual concentration, what is the meaning of the concentration of IgG? Is that really an indication of how severe a response would be, or do we know what that means at this time?
Difficult to say, because we determine at the moment whole IgG, so we do not know which portion or which portion is IgG1, 2, 3, and 4. So I would say no, we cannot conclude from a really high IgG level that at the moment it is severe reaction to the food, because it can all be IgG4 or IgG2, which is also a good guy. So it's difficult to say. Yeah.
Another question is, are we undergoing IVDR with our assay? Where are we with that process?
You're asking if the FOX test will be an IVDR product?
Correct.
Yes, we hope so. We are doing everything for that. We are fighting for that. Yes, we hope. Yeah, but at the moment we are really doing a lot of studies in our R&D department, because we want to really go in deep into the case IgG subclasses. We want to analyze IgG1, 2, 3, and 4 reactivities with a lot of different clinical manifestations, with a huge cohort, because we believe that perhaps not only IgG4 could play a role. We think that also IgG2 could be important for the tolerance induction, and we really want to look at this at the moment. Yeah.
Okay, and another question is, why is less serum needed to run the FOX IgG test than the ALEX IgE test?
Why we use less serum for the FOX test?
Correct.
Yes, we just use 10 microliter for the FOX test compared to 100 microliter for the ALEX, because in the human blood there is much more IgG antibodies compared to IgE, and that's why we can dilute it much more, otherwise the signal would explode. We have to dilute it, so less serum necessary for the FOX test.
Okay, well, if anyone has additional questions, feel free to enter them in the chat box, and we'll get to them once the webinar is complete. I know, Sandra, you mentioned doing some additional testing internally with the different subclasses.
Yes.
But I understand we also have a study, correct, that's coming up with a group in Germany with some IBS patients. Is that correct?
Yes, we are doing studies with Germany and with Switzerland at the moment, because we think that now we are, when Medax established the FOX, they did not know a lot about the different subclasses. So they just said, okay, let's go for IgG testing, so the whole IgG. But I think now we are kind of a time where we have to understand what does the different IgG subclasses mean. IgG4, I totally agree with you. It's a tolerant. It's induced when you do successful allergen-specific immunotherapy. IgG4 is induced. It's the good guy. It's tolerance induction. But I think at the moment it's really a good time to go into research, to take the time and analyze it, and then to just improve food intolerance testing. Yeah.
All right, well, thank you so much, Sandra, and thank you to our attendees for joining us today. We know your time is valuable and hope you found the seminar to be worthwhile. We do have 3 short survey questions that will appear once the webinar closes. We would appreciate your responses. Thank you again, and please look forward to an invitation to a future webinar. Thank you.